In this lecture, we will be covering the molecular basis of inheritance. The molecular basis of inheritance is a 4-part lecture series. In this video, we will learn the history of how DNA was uncovered as the genetic material, as well as the experiments that went along with it. In part 2, we will dive further into that genetic material, DNA, including understanding its structure and the mechanics of base pairing. In part 3, we'll uncover the mechanism of DNA replication and repair, a favorite topic of biology tests everywhere. And in part 4, we will turn our attention to chromosomes, including their components, structure, folding, and my personal favorite, epigenetics, a field still young and emerging and intimately tied to aging and longevity. But for now, let's put our lab coats on, as we'll spend most of this lecture in the science labs of the 19th and 20th centuries. Today, you'll learn about the search for genetic material, how DNA can transform bacteria, how viral DNA can program cells, and the experiments that showed this. We'll also touch on the mechanics of horizontal gene transfer and the structural components of DNA, including an intro to base pairing. Please use the timestamps in the description below if you have something specific that you want to skip to. This molecule hardly requires an introduction. It's arguably the most important cellular component and has relished this role since its discovery in the mid-20th century. We're speaking, of course, about DNA, or deoxyribonucleic acid. In the intricate tapestry of life, DNA is the silent architect, the mastermind behind our existence. It's a spiraling code, a cosmic instruction manual that shapes who we are. But this genetic tapestry is not so easy to unravel, and it was only relatively recently that the intricately woven strings began to unveil themselves. It began in the 19th century when an Austrian monk named Gregor Mendel sowed the seeds of curiosity with his pea plant experiments, unknowingly planting the roots of genetics. With his experiments, he uncovered patterns in the heredity of characteristic traits like flower petal and seed colors, today called Mendelian laws of inheritance. He also determined that there must be heritable units, which we now know of as genes. This work sparked the exploration to identify the units and mechanisms of heredity, and for this reason, Mendel is known as the father of genetics. At the turn of the 20th century, scientists around the world were conducting experiments to find and grasp the threads of life. One of the most important discoveries was chromosomes, the thread like structures within cells. In the 19th century, Swiss scientist Carl Wilhelm von Nageli and others observed them but couldn't decipher their significance. Walter Sutton, an American biologist, and Theodore Boveri, a German scientist, independently unlocked the mystery of chromosomes around 1902. They recognized these structures as the carriers of genetic information, referred to as the chromosome theory of heredity. Sutton's work with grasshoppers revealed the connection between chromosomes and Mendel's inheritance laws, laying the foundation for modern genetics. However, there was still much to be discovered as every advancement led to more questions. What were chromosomes made of? And which of these components is responsible for heredity? Many of these answers came decades later as innovation in technology supported and propelled scientific discovery. Have a look at this timeline. During the mid 20th century, scientists made monumental strides in understanding the nature of DNA and its components. In this lecture, we will learn about and discuss these experiments and how each of them led to modern understanding of genetics, where we can now clone and manipulate the intricate tapestry of DNA. Let's return to the 20th century. There are a few experiments we will focus on in this lecture. First, we'll give a brief overview, then dive into each one. In 1910, Thomas Hunt Morgan's work on Drosophila flies showed that genes are located on chromosomes. The two components of chromosomes, DNA and protein, became candidates for the genetic material. Frederick Griffith's work on bacteria in 1928 showed that pathogenic heritable molecules passed from dead to alive bacteria, and he called these transforming particles. In 1944, Oswald Avery, Colin MacLeod, and Macklin McCarty identified these transforming particles. This event is considered the beginning of molecular genetics. Later, in 1952, Hershey and Chase used radioactive isotope tags to show that DNA is the genetic material of bacteriophages. So, while the role of chromosomes in heredity was discovered using flies, the role of DNA in heredity was first discovered by studying bacteria and the viruses that infect them. Let's begin with Thomas Hunt Morgan's work in 1910, which showed that chromosomes contain the genetic material. For his work, he used the model organism Drosophila melanogaster, a species of fruit fly. Drosophila is typically used in research owing to its rapid life cycle, relatively simple genetics with only 4 pairs of chromosomes, and large number of offspring per generation. On the right, we see a cross for the trait eye color. Notice the two eye colors, red and white. Red is the wild type, meaning this color occurs more frequently in nature and is typically the dominant allele. White, on the other hand, is the mutant color, which occurred at a lower frequency and in an interesting pattern. He began with a parental generation, crossing a white-eyed male with a red-eyed female. After many cycles of breeding, he noticed a few things. 1. All F1 progeny had red eyes. 2. The allele frequency of the red to white colored eyes in the F2 generation was always 3 to 1. And 3. Only the males had white eyes. This 3 to 1 ratio was documented before in Gregor Mendel's work with pea plants. The differences in frequency between the males and females indicated that sex was important for eye color expression. This work uncovered the concept of sex-linked traits, which are genes for certain traits being carried on the X or Y chromosomes, and validated the chromosome theory of heredity. Now, as a concept check, we have a Punnett square analysis of eye color. This cross is different. Here we have a red-eyed male crossed with a white-eyed female. On this, we can see that the genes annotated as X superscript W, with the capital W meaning red eye allele and the lowercase w meaning white eye allele. Here the red is dominant to the white. Notice again that all female progenies have red eyes while the males have white eyes. Why do you think this is? If you need a refresher on Punnett squares, have a look at my lecture on Chapter 15, The Chromosomal Basis of Inheritance. Morgan's work with these tiny insects laid the foundation for modern genetics, earning him a Nobel Prize in 1933, and you can see the prize in the bottom left of the screen. Scientists now understood that chromosomes contained the genetic material. With this discovery, the two components of chromosomes, DNA and protein, became candidates for the genetic material. This is because chromosomes are made of those two components, DNA and proteins. Carefully designed experiments were needed to tease out the minute details of heredity. Scientists were in hot pursuit to figure out which of these made up the genes. Early on, DNA was thought to be the structural component of chromosomes, whereas the genes were thought to be made of protein component of chromosomes. Virus work led many to believe genes were super enzymes. Think about that for a minute. They had it backwards. Protein is now known to be the structural component of both cells and chromosomes. Proteins called histones seen here are used to wrap the DNA and keep it orderly, but they are not the actual genetic material. We'll talk more about this in part 4. But the answer to this question of DNA or protein didn't come until the mid 1900s, along with a new concept called DNA transformation. It began with Frederick Griffith in 1928. Before we get into the experiment details, let's talk about transformation. Transformation is a change in genotype and phenotype due to the uptake and incorporation of foreign DNA from the environment. This occurs through the cell membrane. But let's not overcomplicate this. Simply, it's one organism taking in foreign DNA from its environment. The visual shows this. Now, we all know that the cell membrane plays a crucial role in regulating what can or cannot move into the cell. That's its job. So, transformation isn't something that just happens. Cells aren't bringing foreign objects in willy nilly, especially foreign DNA. For this to happen, cells have to be in a state of competence. Competent cells are those whose membranes have been altered to make them porous, meaning that they have pores or holes in them. This can occur in nature as a time-limited response to environmental conditions like starvation, cell density, or this can be artificially induced in a lab setting using chemicals, heat shock, or electroporation. In both cases, competence leaves the cell membrane porous, which allows it to be transiently permeable to foreign DNA. Again, here we can see the plasmid, which is circular DNA, being taken up by this bacterium. Now that you know what transformation is, let's go over 2 experiments that led to the understanding and discovery of DNA transformation in bacteria. The discovery of the genetic role of DNA began with research by Frederick Griffith in 1928. Griffith worked with 2 strains of S. pneumoniae bacterium, one pathogenic shown in red and one harmless or non-pathogenic shown in green. Have a look at the graph. Notice that the virulent pathogenic red strain has a polysaccharide coating that protects it from its environment, or in this case, from an immune system attacking it. Meanwhile, the green non-pathogenic strain does not have this protective coat, so an immune system can attack and prevent this bacteria from causing sickness. When Griffith injected a mouse with the living pathogenic S strain with that capsule, the mouse died. When he injected a mouse with the living non-pathogenic R strain, which lacks the outer capsule, the mouse did not die. This is what we would expect, right? Now, here comes the experimental part. When you heat these bacteria, you kill them. Remember, heat denatures and unravels proteins and enzymes. So, when Griffith heated the pathogenic S strain, the red one, he killed them. Therefore, when these were injected into the mouse did not die. You can see this in the third panel. However, when Griffith mixed those heat-killed remains of the pathogenic S strain with living cells of the harmless green R strain, some living cells became pathogenic. And when he injected this mix into the mouse died, and living red S cells in capsules were found in the mouse's blood, meaning that these cells could reproduce, producing more S cells. And you can see this in the fourth panel here. So, Griffith observed that pathogenic heritable molecules passed from dead to alive bacteria. And in his incomplete understanding of the role of DNA in genetics, he called these molecules just transforming particles. He called this phenomenon transformation, now defined as a change in genotype and phenotype due to assimilation of foreign DNA. Again, a bacterium takes in DNA from its environment, often DNA that's been shed by other bacteria. This is the first experiment suggesting that bacteria can transfer genetic information. However, scientists still did not know what the genetic material was. Top contenders were proteins and nucleic acids. There are some major differences between the components, shapes, and functions of these two macromolecules. Scientists knew that there were 20 amino acids that make up proteins, while there were only 5 nucleotides that made up DNA and RNA. Further, proteins were capable of many more complex shapes and therefore functions. And for these reasons, scientists believe that DNA was far too simple to be responsible for inheritance. So, we needed some more data, some more experiments to try to figure out which of these were the genetic material. Work by Avery, MacLeod, and McCarty identified the transforming substance as DNA. They isolated the S strain from the dead mice and isolated the proteins and the nucleic acids, the RNA and the DNA, which were the two possible genetic candidates. For their experiment, they used enzymes that specifically degraded each component and then used each mixture separately to transform the R strain. Degrading proteins or RNA had no effect on the virulence of the bacteria, but they found that when DNA was degraded, the resulting mixture was no longer able to transform the bacteria, whereas all the other combinations were able to transform the bacteria. This led them to conclude that DNA was the transforming particle, and this finding is considered the beginning of molecular genetics. Although the experiments of Avery, MacLeod, and McCarty had demonstrated that DNA was the informational component transferred during transformation, DNA was still considered to be too simple of a molecule to carry biological information. Proteins with their 20 different amino acids were regarded as way more likely of candidates. So, the decisive experiment conducted by Martha Chase and Alfred Hershey in 1952 confirmed that DNA was indeed the genetic material and not proteins, and we'll get to that experiment in just a few minutes. First, let's talk just a little bit more about transformation and how this process is used by scientists in the lab. Scientists are always trying to hijack cellular processes and figure out ways to use them for research. With transformation, scientists can make multiple copies of DNA called DNA cloning. They can make large amounts of specific human proteins. For example, human insulin, which can be used to treat people with type 1 diabetes. Virtually all of the insulin used by diabetics worldwide is harvested from transgenic bacteria. So, transformation can be used to genetically modify a bacterium or other cells. Here's a cute example. These pigs were genetically modified with green fluorescent protein, GFP. This gene originally comes from jellyfish, but it's used to help detect transgenic animals. This lets scientists know that transformation was successful. Actually, if transformation is in animal cells, it's called transfection, but it's a similar concept. And I'm sure you've all heard of antibiotic resistance, where bacteria may become resistant to antibiotic treatments. This also can occur via transformation or other horizontal gene transfer HGT mechanisms, which I will mention in the next slide. An antibiotic is an antimicrobial chemical that kills or inhibits the growth of microorganisms. They're used to cure many deadly infections, for example, bubonic plague, diphtheria, and tuberculosis. And if you work in a hospital or healthcare environment, you've probably heard of MRSA, methicillin-resistant Staphylococcus aureus, which is a contagious antibiotic resistant staph infection that can become dangerous. Why? Because it does not respond to our treatments. In this image, you can see transformation at work. In number 1, we can see an antibiotic sensitive bacteria. In step 2, transformation is occurring. A DNA plasmid is being transferred from one environment, in this case, it's another bacterium, to the other. In step 3, the transferred plasmid is being read and the proteins made. And these proteins are genes that make the bacterium antibiotic resistant. And in step 4, we have a transformed bacteria culture, which is antibiotic resistant. So now you have a good understanding of transformation. And just like that, we're about halfway through this video lecture. If you're finding this helpful, I would love if you give this video a thumbs up. I'm all ears for topics and materials you want more of, so please drop a comment below. I also have a Sendowl shop where you can download some of the materials I make, including these very lecture slides. Now, if you don't already have, grab some water because we learn better when our neurons are hydrated. And we still have a few more experiments to discuss. So now you should have a great understanding of transmission. I mentioned that transmission is a method of horizontal gene transfer, HGT. Another name for this is lateral gene transfer. They both indicate sideways movement. HGT is the movement of genetic material between organisms other than by vertical transmission of DNA from parent to offspring via reproduction. So, when we inherit DNA from our parents, that is vertical transmission, and it is via reproduction. Horizontal gene transfer is different. Look at the image. You should recognize these as the 3 domains of life: bacteria, archaea, and eukaryotes. Well, something interesting happens when you look at the DNA of these. We see that some species of eukaryotes have plastids. These are organelles like chloroplast that allow for photosynthesis. And which species are these? Of course, plants have plastids. Well, not all eukaryotes have these. As we know, we don't. Animals don't. So, where did they come from? Well, have a look at bacteria. Many, but not all of them have plastids too. So, what do we see in this image? We see that there was horizontal gene transfer, lateral movement of the genes from one species of bacteria to a species of eukaryotic cell a very long time ago. Now, again, vertical transmission would be transferring of DNA from one generation to its progeny the way that we transfer DNA. And that gene would be moving vertically up the lines of this graph. Again, though, this is horizontal gene transfer, so it's moving laterally. So how does this happen? There are 3 mechanisms of horizontal gene transfer: transformation, conjugation, and transduction. As we know, transformation is the change in genotype and phenotype due to uptake and incorporation of foreign DNA. Conjugation is a little bit different. This is the transfer of genetic material between 2 bacterial cells that are in direct contact. You can see the 2 bacteria connected with pili. Transduction is the injection of foreign DNA by a bacteriophage virus into the host bacterium. This brings us to our next experiment. More evidence of DNA as the genetic material came from studies of viruses that infect bacteria. These viruses are called bacteriophages or phages or bacteriophages or phages. And you can see 2 phages pictured here. We've all experienced the impact that a virus can have, but what exactly is a virus? A virus is DNA, sometimes RNA, enclosed by a protective coat, often simply just a protein coat. We see it labeled as a protein capsid here. Phages have been widely used as tools by researchers in molecular genetics. In 1952, Alfred Hershey and Martha Chase showed that DNA is the genetic material of a phage known as T2. They designed an experiment to show that only one of the two components of phage T2, again, either DNA or protein, enters an E. coli cell during infection. To do this, they used radioactive isotope tags, radioactive phosphorus 32P to label DNA, and radioactive sulfur 35S to label the protein coat. Now let's focus on this for a minute. When we say radioactive, we mean that there are extra neutrons packed into the nuclei of these atoms. This makes them decay and release energy. This allows them to act as a tag, and we can watch where they go. Now, why radioactive phosphorus? Remember, we're trying to figure out if the genetic material is DNA or protein. Phosphorus is abundant in DNA. See the P circled up at the top in that nucleotide? DNA is a polymer of nucleotides, each consisting of a nitrogenous base, a sugar, and of course, a phosphate group. Whereas sulfur is present in two amino acids of protein, specifically cysteine and methionine, pictured here. Find the sulfurs circled in each. So, Hershey and Chase used these radioactive phosphorus and sulfur isotopes to track which of the components of the virus, DNA or protein, was moving from the virus into the bacteria to infect it. Here's the experiment. On the left in blue, Hershey and Chase added the T2 phages into growth medium containing radioactive phosphorus. On the right, the T2 phages were added into growth medium containing radioactive sulfur. The T2 phages incorporated the radioactive materials either into their DNA, shown in blue, or their protein, shown in green. The T2 phages were then allowed to infect bacteria. In step 3, the cultures were collected. We saw step 1 and 2 in the previous slide. So in step 3, the cultures were collected and separated. The scientists only wanted the now infected bacteria. In step 4, the bacterial pellets were cultured and analyzed. Bacteria infected with T2 phage radioactive phosphorus in the DNA produced more 32P labeled phages. On the other hand, bacteria that were infected with radioactive sulfur containing phages produced new phages with no labels. With this, they concluded that the injected DNA of the bacteriophage provides the genetic information. So now, the world finally knew that DNA was the genetic material. Now that we know DNA is the genetic material, let's talk about the composition of DNA. 1950 and the 1940s, a few years before the Hershey-Chase experiment, Erwin Chargaff reported that DNA composition varies from one species to the next. This evidence of molecular diversity among organisms made DNA a more credible candidate for the genetic material. Two findings became known as Chargaff's Rules. 1. The base composition of DNA varies between species, but not within tissues of the same individual or between individuals of the same species. And 2, in any species, the number of A and T bases is equal, and the number of G and C bases is equal. The basis for these rules was not understood until the discovery of the double helix, which of course is the structure of DNA. Let's focus on Chargaff's rules for a minute. Why do they make sense considering what we know today about the structure and function of DNA? It makes sense that different species contain different DNA. Humans and cats and dogs are all different because we have different genes. Look at this chart comparing the DNA content of different species. Of these species, who has the highest percentage of A? Octopus. Do they have an equal percentage of T? You'll notice that it's not the exact same, but it is very close. And the differences could probably be chalked up to experimental error or even some mutations. Now, who has the highest percentage of C? That would be E. coli. And as a brain teaser, look at the bottom organism. What do you notice about these numbers? They don't match. A does not equal T and G does not equal C. And why do you think that is? It's because this species doesn't have two strands of DNA. It's just a single strand of DNA. So it doesn't need to have matching A and T or G and C. For everyone else, the ratio of A over T or G over C is hovering around 1. Now, what about within tissues of the same individual? Within humans, our muscle tissue and our bone tissue and our neural tissue all have the same DNA in the cells. It's just that different genes are expressed in different tissues. And we know this. So his first rule, checks off. Now, what about the second rule? Why are there equal numbers of adenine and thymine? Of course, now 80 years later, we know all about base pairing. We know that A will always pair with T, so it does make sense that there are equal numbers of those. Same goes for cytosine and guanine, which always pair in DNA. And we can see that in the image on the right. A pairs with T, C pairs with G. We also saw it in the previous table. Now, a fun fact about science at this time, many of the big name scientists that fill our textbooks knew each other, or at least crossed paths. In 1952, Chargaff met Watson and Crick and explained his findings to them. Chargaff's findings proved immensely useful as Watson and Crick were getting ready to propose their DNA double helix model. You can see how science builds upon previous discoveries, sometimes in a slow and laborious process. While DNA and genetic engineering are immensely useful and can be life-saving, I like to keep a little perspective. Chargaff warned in his 1978 book, Heraclidian Fire, that, quote, the technology of genetic engineering poses a greater threat to the world than the advent of nuclear technology. An irreversible attack on the biosphere is something so unheard of, so unthinkable to previous generations that I only wish that mine had not been guilty of it. End quote. Just something to ponder. Let's take a break to check your understanding. Match the term in the left column to the correct answer in the right column. Pause the video, then play to check your answers. And if you feel like this has been helpful, please let me know by liking this video. How'd you do? Can you come up with a short explanation of Avery-MacLeod and McCarthy's experiment? Check your answers, and if there are any things that are unclear, look down in the description box for some timestamps to watch that part of the lecture again. We're almost done. To wrap up, let's touch on Watson and Crick and the DNA double helix. In 1953, Watson and Crick unveiled the double helix structure. Chargaff's base pairing rules revealed the complementary nature of DNA strands. These breakthroughs illuminated the building blocks and structure of DNA, revolutionizing genetics. Here are the 4 nitrogenous bases: adenine, thymine, cytosine, and guanine in their complementary base pairings. In the next part, we will learn all about the DNA structure and the double helix. We'll also dive into some history of how it was discovered. And there you have it, a brief highlight of the greatest discoveries and advances in DNA from the 19th and 20th centuries. From Gregor Mendel first noticing patterns of inheritance in pea plants to scientists being able to clone a mammal, we've come so far. In part 2, we will dive into DNA structure and base pairing. In part 3, we will cover DNA replication and repair. In part 4, we will cover chromosomes and packing, along with some epigenetics. Thank you so much for watching, and I will see you next lecture.